Next class, previous class we discussed about the spread method. Then today this is about the topic special method of isolation of pure cultures, single cell isolation method, capillary peptide method. Several small drops of the suitably diluted culture media are put on the sterile glass cover slip by a sterile peptide down to a capillary. Once the examinations, each drop under the microscope until one finds such a drop which contains only one microorganism. This drop is removed with a sterile capillary peptide to fresh media. The individual microorganism present in the drop start multiplying to yield a pure culture. Capillary method for obtaining a single microbial cells. Single microbial cells. Droplets containing microorganisms are put on the sterility cover strip with the capillary peptide. Droplet containing only one microbe is picked up by the capillary peptide. Each droplet is examined under microscopic until a droplet containing only one microbe is found. The droplet is transferred into a culture tube containing suitable media. Micromanipulator method. Micromanipulator have been developed which permit one to pick out a single cell from a mixed culture. This instrument is used in conditions with a microscope to pick a single cells, particularly bacterial cells, from a hygienic drop preparations. Advantages of manipulation method. The disadvantages of this method are that one can be reasonably sure that the cultures come from a single cell and one can obtain stains with the species. Disadvantages are that equipment is expensive. Manipulation is very treacherous and it requires a skilled operator. And picture samples of slide glass, microcapillary water sources, springing gum, objective lens. Enhancement culture method. Generally, it is used to isolate these microorganisms which are present in a relatively small number of that have slow growth rates compared to the odd spaces present in the mixed culture. The enhancement culture stages provides a specially designed cultural environment by incorporating specific nutrients in the media and modifying the physical conditions of the inoculations. The medium of non-compositions and specific conditions of incubations favor the growth of described microorganisms but is unsuitable for the growth of other types of microorganisms. Generally, it is used to isolate dissolved microorganisms which are present in the relatively small number of that have slow growth rates compared to other species present in the mixed culture. Maintenance and preventations of the pure culture. When a microorganism has been isolated and grown in a pure culture, it becomes necessary to maintain the purity of the microorganism by keeping the pure culture free from the contaminations. Once a microorganism has been isolated and grown in pure culture, it becomes necessary to maintain the purity of the microorganism by keeping the pure culture free from the contaminations. Normally, in laboratories, the pure cultures are transferred periodically into a fresh medium subculture to allow continuous growth and vitality of microorganisms. The transfer is always subject to aseptic conditions to avoid contaminations. Maintenance and preventations of pure culture. The transfer is always subject to accept conditions to avoid contaminations. Normally, in laboratories, the pure cultures are transferred into a fresh medium and a subculture to allow continuous growth and vitality of microorganisms. Objectives of preventations. to maintain isolated pure cultures for ended periods, future uses in a valuable conditions, to avoid the contamination, to restrict genetic changes, mutations. To maintain isolated pure cultures for extended periods of future use in a variable conditions, to avoid the contaminations, to restrict genetic changes, mutations. Objectives of the preservations. Then objective and preservations to maintain isolated pure cultures for extended period and allow future users in a valuable conditions, to avoid the contaminations, to restrict genetic changes and mutations. Then next fourth topic, applications of fermentations, academic use, research purpose, fermentation industry and biotechnological field. Academic use, research purpose, fermentation industry, biotechnical field. Once a microorganism has been isolated and grown in a pure culture, it becomes necessary to maintain the validity and purity of the microorganism by keeping the pure culture free from the contamination. Normally in laboratories, the pure cultures are transferred periodically onto or into a fresh medium, subculturing, to allow continuous grow and vitality of microorganisms. The transfer is always subject as aseptic conditions to avoid contaminations. Since repeated subculturing is a time consuming, it becomes difficult to maintain a large number of pure culture successfully for a long time. In addition, there is a risk of genetic changes as well as contaminations. Therefore, it is now beginning replaced by some modern methods that do not need frequently subculturing. These methods include refrigeration, paraffin method, and lipophosis, അല്ലെങ്കിൽ freezing drying. These methods include refrigeration, paraffin method, and freezing drying. Then methods of preservation. Periodic transfer to fresh mediums and subculturing, storage in a sterility soil, storage at low temperature, presentation by overlying cultures with the mineral oils, freezing and drying, lipophosphations, periodic transfer to fresh medium, subculturing, storage in a sterility soils. Periodic transfer to fresh medium. Strains can be maintained by periodically preparing a fresh culture from the previous stored cultures. The culture medium, the storage temperature, and the time interval at which the transfer are made vary with the species and must be instructed behold. The temperature and the type of medium chosen should support a slow rather than a rapid rate of growth, so that the time interval between transfer can be as long as possible. Many of the more common heterotrophs remaining variable for several weeks or months on a medium like a nutrient occur. The transfer method has the disadvantage of failing to prevent changes in the characteristics of strain due to the development variant and mutants. Advantages: it is a simple method, any special apparatus are not required, easy to recover the culture. Disadvantages: risk of the communications is more. It may be possible to change the genetic and biochemical characteristics, statics. In storage in a sterility soil, it is mainly applied for the preservation of synthetic microorganism, a single store within a cells. Single storage involves incubation of 1 ml of spore suspension into soil, autoclaved twice, and incubating at a room temperature for 5 to 10 days. The initial growth periods allows the fungus to use the available moisture and gradually to become a dormant. The bottles are then stored at a refrigerator. Validity of organism found around 70 to 18 years. Storage of to low temperature. Cultural media can be successfully stored in the refrigerator or cold room when the temperature is maintained at 4 degrees Celsius. Another liquid nitrogen can be provided long-term presentations of culture. In this method, different suspensions of microbials is prepared in a medium containing a protective agent. At the temperature range, the metabolic activities of microbes slow down gently and only small quantities of nutrient will be utilized. This method cannot be used for a very long time because toxic product get accumulated which can be killed the microbes 10 to 13 years without changing the characteristics. Preparations by overlaying culture with mineral oil or liquid paraffin storage. In this method, sterility liquid paraffin is produced over the slant culture of microbes and stored upright at room temperature, whereas the cultures can also be maintained by covering agar slant by sterility mineral oils, which is stored at room temperature rated properly at 0.5 degrees Celsius. It limits the oxygen access that reduces the microorganism metabolic and growth as well as to cell tiring during presentations. Advantages: simple, mainly used anaerobic microorganism, cost-effective method, can refer 10 to 15 years. The freezing drying. Freezing drying is a process where water and other solvents are removed from a freezing product by some limitations. Then pictures: small cotton punctured vials containing foraging suspensions of the microorganism are placed in the glass flask which is attached to a condenser. The condenser is conducted with a high vacuum pump and the system beginning about decisions of the cultures. Procedure: in this process, a dense cell suspension is placed in a small visual and foraged at 60 to 70 degrees Celsius. The vial are immediately conducted to a high vacuum liner. The ice present in the foraging suspensions. Advantages: only mineral storage spaces is required. 100 of liquefied cultures can be stored in a small area, remained viable for more than 13 years. Frequently, subculturing is not required, maintained without contaminations. Small visuals can be sent conveniently through the mail to the microbiology laboratories when packed in the special sealed mailing containers. This method is employed for the presentations of serotoxins, enzymes, and other biologicals. Conclusion: pure culture maintenance techniques is used for the isolations and presentations of culture from difficult sampled. These techniques are used to industrially important organisms. It is external to check the quantity of the provide organism stocks. However, presentations is external as the reversible. Great importance is field of science. Presentations help in research purposes. Industry also as the microbiological field. Reference.